Thanks, Cole. It was a good introduction to the event that we all collaborated on last fall. And I just want to point out that most of this talk was put together over the last couple of months while I was down in Antarctica. And the primary authors on this were Kate Hubbard from Florida Fish and Wildlife and Steve Archer, who works and manages our Bigelow Analytical Services Lab that does a lot of the toxin testing in collaboration with DMR. So the response to the big bloom event last fall was a pretty unique collaboration among a huge number of different agencies and institutions, and it was really actually encouraging. I've been, as an independent researcher at Bigelow Lab, I've been trying to get funding from the federal government to study pseudonychia for the last 4 or 5 years. And what it really boils down to is that there's not a lot of money available at the federal level for harmful algal bloom research. I think the year that we came very close to getting it, there might have been $2 million set aside for the entire country. So that was a little bit sobering to realize that. Oops.
You just jiggle that cord on the left. It should come back.
Okay. Oh, there we go. Okay, so in response to the high Domoic Acid numbers that DMR was seeing, NOAA released a small amount of money that was divided between Woods Hole Oceanographic and Bigelow Lab to mount a rapid response during the bloom. And so this is really a unique situation where we ramped up in the matter of about a week to respond to sampling, to respond to these high numbers of Domoic acid that we were seeing in the field. Of course, these blooms just don't come out of nowhere. Typically, there's some environmental conditions that have to be just right to lead to the bloom. As Cole mentioned, the blooms of pseudonychia are a regular event in the Gulf of Maine. Occasionally, these blooms will go toxic, and there'll be Domoic acid production. And then the Domoic acid gets passed into the food web, starting with the smaller consumers of the phytoplankton, and then can actually get passed up the food chain via consumption by top-level predators of the lower-level consumers. I come to pseudonychia research after spending about 10 years in Southern California, where we had these ASP events every single year. They were very predictable. You could almost set your calendar to them. And we would have very high abundance of pseudonychia. There's a lot of upwelling along the Southern California coast, so there were nutrient-rich waters for most of the year. And I lived about just a mile away from this facility that would treat sea lions that would essentially wash up on the beaches and interact with the thousands of beachgoers. And the animals, the marine mammals, would become disoriented. They would start, acting confused. They would abandon their pups. And we even had cases of pelicans landing on the 110 freeway, which goes right into downtown Los Angeles. So the event, around here, we don't necessarily see this kind of response to a toxic algal bloom, but it was very evident in Southern California. We knew that we did have regular blooms here when I moved to Maine about 7 years ago. And so I started just trying to track the regular blooms of pseudonychia down in the Booth Bay region. So in response to the most recent event, very quickly a grid was set up along the mid-coast to down east Maine, and about 24 stations were monitored. The idea was to look at the spatial extent of the bloom through a variety of sampling techniques, to look at both the toxicity and the species composition. As Cole mentioned, it's not easy to just look at a sample and figure out what species you have, and then actually try to validate the detection tools, because as you might imagine, the tools are changing rapidly. There's a variety of techniques for looking at the actual toxin itself, and for identifying the species in a more high throughput manner than microscopy allows us to do. So we went out and we actually split up this sampling between a Woods Hole group that chartered the Gulf Challenger from the University of New Hampshire, and then we took one of our vessels and trailered it up to the down east sites where Gulf Challenger couldn't make it in the amount of time that we had, and then we wound up chartering a boat for a second day of sampling. I'll say that I wasn't on the boat. We sent a couple of people that were able to break away from their daily routines for that sampling. We had this sampling also, like I mentioned, allowed us to do this intercomparison via our microscopy, our scanning electron microscopy, our regular light microscopy, and then a variety of genetic tools that I'll get into in a few slides. So what did we find out? This is a graph that shows pseudonychia, the domoic acid, and then pseudonychia abundance in cells per liter at a number of the stations. And you'll see that, so there's a scale from 0 to about 15 micrograms per liter. And this is particulate domoic acid. So this is the domoic acid that's in the phytoplankton, in the pseudonychia cells that we were able to collect on eFilter. The number that Cole was referencing, 20 micrograms per gram, is grams of shellfish tissue. I don't think, and Cole, you can correct me if I'm wrong here, that there's a standard for domoic acid in the plankton. Okay, so that has not been established yet. But what we do know, by comparing these numbers to other numbers from the west coast of California in particular, is that once you're getting up around 12 to 15, these are pretty high numbers for domoic acid in the plankton. And you can see that the greens, the yellows, and the reds were all concentrated, and I believe the map is sort of crude here, but I think this is the Jonesport to Machiasport area of the coastline. So that's where the highest numbers seem to occur. Many of the other stations had relatively low domoic acid in the plankton. With respect to cell numbers, as Cole mentioned, there didn't seem to be a good correlation between cell abundance and toxicity. So as you can notice here, there's some orange to yellow values. This is above 150,000 cells per liter. And I think your numbers, Cole, were they cells per liter or cells per liter? Okay. So that is the same comparison. So these were very, very high cell numbers, kind of unlike anything we've seen in the last few years. Pseudonychia is this chain-forming diatom. It's important to remember that diatoms are made of elemental silica, so when they die, they're heavy, they sink out of the water column, so a bloom can come and go very quickly. It can sediment out of the water column very quickly. Up to 14 different species have been described for the Gulf of Maine, and many of these do have the ability to produce the domoic acid. This is a sample, a picture that I took, just a light micrograph from one of the hotspots of domoic acid production. I wasn't actually able to, one of the things we were trying to do is get these cells in culture in the laboratory so that we can begin to conduct more experimental investigations on them. I wasn't able to actually cultivate this one, but I did get some cultures started from down in the Booth Bay area. So one of the big discoveries from this sampling event is, for the first time, this Pseudonychia australis was detected in the Gulf of Maine. And this detection, again, you can't just look through a light microscope. You have to use scanning electron microscopy. So this is sort of the maximum magnification technique that you could use to look at a cell. And to identify it to a species level, it relies on looking at the pattern of these silica skeletons. So there are very few people that actually have this experience. This is sort of a dying breed of taxonomists, as most of us are turning more to the genetic techniques. But right now, for better or worse, Pseudonychia is identified by this ultrastructure of the silica skeleton. And we knew that this double row of these pores within one of these channels of striae, or stria, indicated that it was Pseudonychia australis. And this is a known toxic species of Pseudonychia. So that was pretty interesting. I'll spend a few minutes to talk about some of the genetic techniques that are being used and that we're developing. This is a method, the first method I'll talk about relies on amplifying a short segment of DNA within the genome of Pseudonychia and looking at the length variations between the different species. So we're targeting a specific area that we know is hypervariable with respect to its length. And when we do that, we use a process called polymerase chain reaction, where we can target that one species, or with that one section of the genome, and then based on the relative abundance of these peaks, we can generate graphs that look like this, that show you the relative proportion of the different types of Pseudonychia that might be in a water sample. So very briefly, what this is showing you is the length variation of this stretch of DNA. And just because we're targeting this hypervariable area, we know that this particular peak of 168 base pairs belongs to this type 4, which is Pseudonychia delicatissima. Again, each one of these different fragment sizes corresponds to a different species. So it's a very rapid technique to get a relative abundance of the types of Pseudonychia you might have in a single water sample. And there's really, there's no way you could do this via microscopy for the number of samples that we collected just even in this small rapid survey. So increasingly we're starting to rely on these molecular technologies. And this is Kate Hubbard's work from down in Florida Fish and Wildlife. Okay, so as I mentioned, we broke the sampling up into sort of a southern region and a northern region, and at this point we only have about 7 or 8 different stations fully analyzed via this DNA fingerprinting method. So as many as 9 different Pseudonychia were detected, and then these pie charts show the relative abundance of the different types. Interestingly, this bloom seems to be happening all the way from Maine down into Rhode Island at the same time. So it's a fairly large areal extent of the bloom. But to the north, it seemed like we had a higher proportion of the Pseudonychia australis, where to the south, it seemed to be dominated by the Pseudonychia pungens, which is a type we see along the coast of Maine pretty regularly, but it did not seem to be one of the major players in the most recent bloom event, except in this more northerly site. So this is kind of interesting. We had dominance sort of at the northernmost region and then the southernmost region by pungens, but in the middle it seemed to be australis that was the big player. Okay, let me tell you a little bit more about technology that I use a lot in my lab. This is called quantitative PCR. So again, it's relying on amplifying a diagnostic region of the Pseudonychia genome on this type of machinery here. And the interesting thing about it is that this is a lab-based system that costs maybe $30,000 or $40,000. This is a little miniature version of the same technology. And I'm collaborating with a company down in the Philadelphia area that is trying to develop these for use in many different markets and industries. And so I'm one of the beta testers for this, and I'll just very quickly, the sensitivity on this little handheld machine, which is driven by an iPhone that's embedded in it and runs on an app, is about the same as the $40,000 machine. This is still a prototype now, but they're hoping to get this into sort of consumer level markets in the next year or two. PCR, again, as I mentioned, relies on amplifying one target area of the genome, and then through subsequent cycles, you get exponential amplification of that one small region. And when you analyze it on one of these quantitative PCR machines that I just showed you pictures of, the amplification curves look something like this. So you can imagine this is cycle number, which is sort of a proxy for time. And as you run this reaction over and over again, you get more and more DNA amplifying. And in the top curve, the top graphic here shows you a calibration curve. And so we can run known amounts of DNA from the different organisms that we're interested in this case, the Pseudonychia. And so you can run a standard curve ranging from billions of copies of the gene that you're targeting down to just a few copies. It's incredibly powerful technique. And then you can see that it's a very linear relationship between the copy number and then the cycle number at which the DNA crosses this threshold. Okay, so just the take home here is that it's incredibly sensitive. We can detect just a few copies, which is essentially less than one cell in a liter of seawater. And you'd never see that if you were relying on microscopy. So the advantage here is that we should be able to see blooms coming much sooner than we would if we were just relying on microscopy. As I mentioned, we are also trying to cultivate the organism so that we can do experiments with it in the lab. These are a couple of isolates that we were able to grow in the lab as far back as 2012. And another thing that you can do with the quantitative PCR is that after you've amplified it on one of those two machines that I just showed you, you can melt the DNA. And this graphic, I won't go into the nitty-gritty, but what it's showing is that the DNA from the two different species that I was able to grow in the lab falls apart at slightly different temperatures. So you can use this melting temperature of this short strand of DNA as another diagnostic. So again, as far as developing rapid diagnostic tools, we're making some advances in that area. So we should be able to see the blooms coming earlier than we normally would, and we should be able to genotype them much more quickly than we would. The thing I didn't mention about the electron microscopy is that's fairly labor intensive, and again, it relies on taxonomic expertise. This is a survey that one of my students from Colby College did in the Damascotta River a few years ago using this quantitative PCR technology. So she collected samples on 3 different dates at 4 stations along the Damascotta, and within one afternoon she was able to run all the samples on the quantitative PCR machine and determine where the hotspots were for Pseudonychia. In this case, it was the earliest sample, sort of mid-October sample, and it was actually the offshore station that seemed to have the highest concentration of Pseudonychia cells. I'll just point out, we don't know yet what genes are involved with the production of Domoic acid. That would be more interesting to be able to develop a quantitative PCR assay for the genes that were turning on and off the production of Domoic acid. But right now, these are limited to detecting cell abundance, essentially. Okay, so conclusions. We have a new species of Pseudonychia here in the Gulf of Maine. We don't know if it's here to stay. We don't know how closely it's related to the Pacific or Western European strains, but Pseudonychia is a species that's globally distributed. I saw it in Antarctica a couple months ago. It's everywhere. The question is what turns on the toxin production? There's been a lot of work around this, and there's some evidence to suggest that when the cells become nutrient limited, they may start producing the toxins. It might be a deterrent for grazers, so things that would be normally eating the Pseudonychia might be deterred by the domoic acid production. It may also just be a metabolic byproduct that helps them scavenge trace metals from the environment, which they need for their growth. This was a really unique event, but it was a really good test for the scientific community to collaborate together. This is the first time we've worked with Woods Hole on studying Pseudonychia in the Gulf of Maine. I think we'll continue these collaborations. I know we're already talking with Kate down in Florida and DMR for future work. ultimately we want to be able to see these blooms coming. It's not like at this stage, there's not much we can do to prevent them, but if we can know when they're coming, so it might help to advise the fisheries community on best timing, best practices, timing of when to collect, when to pull in shellfish that you might have depurating, or if you're a grower, when to bring that in. And then, again, we're working on our methodology to make sure that we're all speaking the same methodological languages. And we, of course, we'd love to be able to get out there and do greater levels of monitoring. And it's possible that these little handheld PCR devices, as they become more and more available, could be deployed along the entire coastline and allow both citizen scientists and stakeholders to actually do some really powerful testing at the genetic level. So, that's what I have for now. And unless they have questions. Sure.
What does Canada do?
How is Canada?
Do they respond, or do they use the same thing I do?
I think they're probably using standardized methodology, similar to what. So far, I haven't been talking to any of the Canadian scientists.
They had closures last year on the Bay of Fundy for ASP closure. They're not doing any phytoplankton testing right now, as far as I know.
Yeah.
They've lost their phytoplankton receptors. So they're just testing shellfish.
One other comment I make is that actually a couple of us from Bigelow just got an email from one of our colleagues about half an hour ago letting us know that Rhode Island just enacted a pseudonichia closure yesterday. So this just came out for the lower Narragansett apparently. So it looks like it's back, at least in the southern regions, and that's pretty unexpected. So.
Can you not invite him next week?
Okay.
Sure.
The California thing, you said you could see it on the calendar. Was there any water temperature, or was there anything associated with the
The blooms, from what I remember in California, they would always occur sort of mid to late summer. There did seem to be a bit of a temperature component to it. It's not one of, it's typically, just having said that there's a bloom in Rhode Island now,
Yeah.
typically it's not an early season bloomer. It comes in after many of the other phytoplankton blooms. So.
It's an algae. Does photoperiod everything to do with it?
I'm sure there's a lot of research that's been done about competitive. There's a niche at a certain time of the year where this type of cell does better. Normally diatoms tend to bloom earlier in the season. This one seems to come in later. I had a student a year or two ago that wanted to work on pseudonichia, showed up in June. We never saw it. He had to change his project. So it comes and goes. It's not necessarily super predictable. Sure, Dana, with that.
Pete, does Domoic acid have different epimers, and are they produced by different species, and do they have similar or different?
As far as I know, it's one molecule. It's not like the microcysteines that cyanobacteria produce. That there are dozens of different analogs that all have about the same effect. The problem with pseudonichia, with Domoic acid, is that it looks a lot like glutamic acid, and it interferes with nerve cell communications. So to a cell, to a mammal, it looks like glutamic acid. It interferes with nerve cells communicating with one another. So that response that we would see in the seals and sea lions in Southern California was just general confusion. They would stop eating. They had to be taken into these facilities to depurate their digestive system of the pseudonichia and the Domoic acid.
Domoic acid was sort of like discovered in the 80s, I think it was 89, 2009 or something like that in Canada, it killed 3 people and made 250 people sick. So some people had permanent brain loss. It causes lesions to your brain, or can, at high levels, so it's not something to mess around with.
And finally, I'll just make one more comment too, that at Bigelow, I mean, we don't have a solution for it right now, but we do have a process. It's called Centers for Venture Research, by which we will work with stakeholders to try to link up the expertise that we have at the lab with problems that you might be having in the fishery industry or aquaculture industry with harmful algae. So, and I'll direct you to Nicole Price, who's sitting in the back, my colleague, who is directing our shellfish and essentially seafood safety center for venture research. We have time for one more?
Yeah, I think until they come in and actually
Okay,
kick us
so
out.
drag us out, we'll keep going. Sure.
Given that your assaying is relatively new, do you suspect any false positive stuff going forward? We had the, what was it, MSP in the spring, and that didn't...
The DSP.
Yeah.
Yep.
Okay. Well, the genetic techniques are very similar, but they're only as good as the genetic databases, which are improving, by leaps and bounds on a daily, if not weekly, if not daily basis. One thing, where quantitative PCR is one of my primary research tools, I've been burned in the past by bad methods in the literature, so I'm actually a bit of a stickler for designing these assays and making sure that they only hit the organism that you're interested in or the gene that you're interested in. And we use very sophisticated software to develop these assays. So increasingly, I would say that I'm comfortable with, if I get a positive hit, it's because there's pseudonychia there, or it's because the gene I'm targeting is there. And we do test this. We have this massive culture collection at Bigelow Lab, which you may know about, 3,000 different algal strains. So the standard practice is to design a quantitative assay, but then test it against either real DNA samples or to simulate the reaction on a computer and make sure that it's as specific as we want it to be. No, it's a good question.
The failure with the DSP test, it was a totally different kind of test. It wasn't genetic.
I just was curious.
Yeah.
It's always a question, though. You want to be sure if you're saying it's there, or the gene's there, or the toxin's there.
I'd rather have the people not get sick.
Or if you're making closures.
Yeah, no, there's a lot at stake, obviously. Yeah.
This one that has the relation with not, say, raining from July till September.
That's a great question. I'll go out a little bit on a limb here, so don't hold me to this, but my colleague Steve Archer, who is one of the primary authors of the talk in this work, did find a bit of information suggesting that a previous bloom event, not in the Gulf of Maine, was linked to a dry spell followed by a very wet period. Because I think, if I'm remembering correctly, we did have some heavy rains sort of at the beginning of the fall period, maybe like soon before this event started. I don't know. We don't necessarily know yet, but there may be an interesting correlation there. Sort of warm, dry period, then followed by a wet period. Right after a very dry winter. No, that's a good question.
Thanks for sticking it out with us. It's a little past 4. Everybody enjoy the rest of the forum.